Good morning, everyone. Good morning. Good to see you. Happy Monday. Yeah, as always, go ahead and type your name into the chat for me. Thank you very much. I appreciate that. I hope everybody had a nice weekend. I know it was a busy one. Looks like everybody got their work in for me, which is wonderful. Good job. Today, we're going to be looking at a particular type of biochemical testing that we use in the microbiology lab, which looks at carbohydrate catabolism, looks at the ways that some microbes break down or catabolize certain carbohydrates and whether or not they have that ability. And of course, when it comes to biochemical reactions or metabolic reactions, it all depends on enzymes, whether or not the organism has the enzyme necessary to do that particular reaction. So that's what we'll be looking at today. It'll kind of be a carbohydrate week because on Wednesday, we're going to look at another type of biochemical test that examines fermentation ability and whether or not microbes have the ability to ferment just generally. If you remember, when we looked at our selective and differential media, we said we often use the ability to perform a certain reaction as a way to differentiate between organisms. Remember on MSA, mannitol salt agar, we looked at whether or not the staff could ferment mannitol. And in the EMB agar, we asked whether or not microbes were capable of fermenting lactose or sucrose. So we're always asking questions about what these microbes can and cannot do. And basically that's asking whether or not they have the enzymes necessary to perform those reactions. So over the next couple of weeks, as we finish up, we're going to be looking at these types of biochemical tests, a handful of the most common biochemical tests that can help us identify microbes. Now, before I get started into the lab material, let me ask if anyone has any questions about lecture material from last week, the information about energy and enzymes, about cellular respiration, oxidative phosphorylation, any of the lecture material or other material that we've been studying. Go ahead and type it into chat if you do have a question. I also wanted to mention that I sent out an announcement about just some interesting microbiology in the news. I sent out an announcement that there's a post on one of our discussion boards that I put up about Justin Bieber. Justin Bieber's been in the news. And there's a tie in with microbiology because Mr. Bieber is suffering with a type of facial paralysis called Ramsay-Hunt syndrome. And there's actually a tie in to microbiology with this disorder. So if you're interested, I posted that on the discussion board called Microbiology in the News. And just this morning, I pinned that discussion board, so you should be able to find it more easily. All right, it should be right up there with the other 2 discussion boards that we have. So yeah, you find stories about microbiology in very strange places sometimes. All right. So I'm going to just pull up this week's module just so we can have a look. We are rapidly coming to the end of the summer. So I want to make sure that we're always thinking about what's coming next. Here we are in week 8. Here's our to-do list. This week in lecture, we're moving forward again into molecular subjects. We're going to be looking at replication and mutations, and then transcription and translation. This week, there's only 1 lecture quiz. That's an important thing to note. So the lecture quiz will cover both lecture topics. It'll be a little bit longer than the typical lecture quiz because it does cover both topics, and it will be due by Friday at midnight. Here are our 2 lab topics for the week. Again, carbohydrate catabolism today, fermentation on Wednesday, and lab homework due by Sunday at midnight, which is pretty typical for us. Now, looking ahead to next week, I'm just going to scroll down to week 9. Looking ahead to next week, you have your next lecture exam. Now, I wanted to mention this morning because this exam falls on an unusual schedule for us. So far, our first 3 exams have opened up on a Thursday and been due by Sunday. This lecture exam 4 is different. This one opens up on Monday and then is due by Wednesday at midnight. Okay, and you'll see that right here. It opens up at 8 a.m. on Monday, the 18th, and it's due by Wednesday at midnight. Otherwise, you're going to notice that the exam is very similar to previous exams in terms of its length and so on. And then after that exam, there's only 1 topic, Introduction to Immunology. In laboratory, we're moving on to protein catabolism and our final lab topic, which is respiration. This is particularly about anaerobic cellular respiration. So next week, we begin our final unit in lecture, and you're going to see that we're really moving away from the microbes themselves, and we're moving into really public health in this last combination of lectures, this last unit. We're going to learn a little bit about immunology. We're going to learn a little bit about epidemiology, a little bit about vaccination, and finally, a little bit about infectious disease, how pathogens work in terms of being virulent or able to cause disease, and how that disease affects us, how we actually develop these infections and how they progress across time. Most students tell me that they really enjoy this last unit, that it's information that they find very interesting. So I hope that holds true for all of us this summer as well. But we are rapidly coming to the end of the semester, the end of the summer term, I'll call it. So just keep that in mind as you finish up. You all have been working really hard this summer, so don't let up. Don't let up now. Keep pushing, and you'll get a nice grade that you deserve. All right, I'm going to go ahead and pull up our slides for today. That's what you should be seeing on your screen right now. The title slide that says Lab Carbohydrate Catabolism. Now, our objectives for today Our first to define terminology that's related to carbohydrates and carbohydrate catabolism. We've talked a little bit about this in lecture, but we're going to focus in and review terminology that we use when we discuss carbohydrates. We're going to talk about the difference between oxidative catabolism and fermentative catabolism. And for our virtual experiment, we're going to perform two different tests. One is called the Starch hydrolysis test, and the other one is called the OF test. And OF stands for oxidative fermentative. But let's begin with some vocabulary terms. Remember, in science, when we use the word sugar, we're not talking about table sugar. We're not talking about sucrose. We're talking about carbohydrates. We're talking about the macromolecule that is the carbohydrate. Now, when we talk about these sugars, we also use words like monosaccharide, disaccharide, polysaccharide. If you translate those terms, this saccharide portion stands for sugar. And then mono or di or poly tells us how many individual sugars are in that molecule. So, for example, a monosaccharide, we would translate this as one sugar. So these are also what we sometimes call simple sugars. These are single units of carbohydrate. Of course, glucose is the most well-known of the monosaccharides, but there are others. There's galactose and other maltose and dextrose and things like that. Those are all considered simple sugars. I have a term here that you should be familiar with, and that's dextrose. Dextrose is just another name for glucose. So if you ever turn, for example, a container, a food container around and you read the ingredient label, if you see the word dextrose in the ingredients, they're talking about glucose. So glucose is really sort of the granddaddy of monosaccharides. And the most important thing for us to remember about glucose is that it is the universal carbohydrate across all different types of cells. It's the universal carbohydrate that is used to produce ATP in cellular respiration. So it's glucose that bacteria and archaea and eukarya are burning or oxidizing or catabolizing in order to make their ATP. Glucose is, again, the base of many larger carbohydrates. So we see it quite a bit. Remember that plants, green plants, and photosynthetic microbes are able to make their own glucose through photosynthesis. You and I are different. You and I have to consume our glucose. We have to eat plants and eat other organisms in order to get our glucose. So the monosaccharide is the one unit sugar. The disaccharide is simply two monosaccharides that are bound together. And one of those is often glucose. So I mentioned lactose, which is milk sugar, sucrose, which is table sugar, maltose is another very common one in nature. These sugars have two monosaccharides that are bound together. So when we use the term simple sugar in science, we're talking about either a monosaccharide or a disaccharide, one or two units in the carbohydrate. Now, when you hear the term oligosaccharide, oligo translates as few. And by definition, an oligosaccharide has between 3 and 20 individual monosaccharides in it, and they're all bound together. A polysaccharide, poly translates as many, and we define many as over 20 individual units of sugar that are all bound together. That's a polysaccharide. We touched on a couple of polysaccharides earlier in the course. Remember, starch is a polysaccharide that is built from many, many individual glucose molecules, all bound together. Starch is made by plants as a way to store glucose for energy purposes. In animals, we have a very similar molecule called glycogen. Glycogen is also composed of many individual glucose molecules all bound together. And in an animal, glycogen is a storage form for energy. So what I'm saying when I say it's a way to store glucose for energy purposes, what I'm saying is that starch and glycogen are used as a place to put glucose so that when the cell or the organism needs to make some ATP, it can go to the glucose in the starch. It can go to the glucose in the glycogen, take off some of the glucose and burn it or catabolize it to make ATP. Remember, there are other polysaccharides. There are lots of them. Remember, peptidoglycan is a polysaccharide, and that makes up the cell wall around bacteria. Remember, chitin. Chitin makes up the cell wall in the fungi we've talked about. There are lots of polysaccharides. There are some polysaccharides that are not used as a source of storage of glucose for energy, but rather as structural components. Cellulose is a good example of that. Cellulose is also made from lots and lots of glucose molecules. But a plant doesn't make cellulose to store glucose. A plant makes cellulose in order to keep its cells rigid and strong, in order to build stems and branches and leaves. It's not going to go to the cellulose and take glucose out when it needs to make ATP. It's not going to do that. It's going to go to the starch for that. So many different forms of carbohydrate and different uses for that carbohydrate, including storing glucose in order to make ATP. The only other term that I'm going to mention is this term complex carbohydrate. This is another term for a polysaccharide. Now, this terminology is kind of going out of favor, but when I was an undergraduate student, we used to talk about complex carbohydrates instead of polysaccharides. The term polysaccharide is much more common now. Now, microbial cells have lots and lots and lots of what are called hydrolytic enzymes. And they use those hydrolytic enzymes in order to digest carbohydrate that they might encounter in the environment. So hydrolytic translates as water breaking. In other words, they use water to break apart these carbohydrates. They use the enzyme and a little bit of water in order to break apart the bonds in these carbohydrates. Hydrolytic enzymes fall into two groups. There are some that are called exoenzymes, and there are some that are called endoenzymes. A hydrolytic exoenzyme is secreted out of the microbial cell and into the surrounding environment. And outside of the cell, what it's going to do is break down any carbohydrate that might be present out in the environment. A great example of this is the enzyme called amylase. Amylase is an exoenzyme that breaks down starch. So if a microbial cell, like a bacterial cell, is able to make amylase, what it's going to do is make it and secrete it. It's going to send it outside of itself into the environment. And if there's any starch out there, the amylase is going to break that starch down. Remember, starch is just lots and lots of glucose molecules that the plant has tucked away in case it needs the glucose to make ATP. Well, the microbial cell can also use that glucose. So it breaks the starch down, and then it simply absorbs the glucose back into the cell. And it can use that glucose to make its own ATP. That's amylase. Now, there are also hydrolytic enzymes that function as endoenzymes. Remember, exo means out and endo means in. So an endoenzyme is kept inside the microbial cell, and it's used to digest molecules inside the cell. Now, let me just adjust this a little bit. One of the tests that we'll be looking at today is a test called the Starch hydrolysis test. And what this test is going to look for is whether or not the microbe, the bacterial cell, makes and secretes this exoenzyme called amylase. We use a medium shown over here in the A field. We use a culture medium that has starch in it. We put starch into this medium, and then we streak our microbial cells onto the plate and incubate it. And we're going to look to see if that starch has been broken down. So after incubation is over, we use this to tell us what has gone on. This is just iodine. We add some iodine to the plate after incubation. And what's going to happen is anywhere that there is starch remaining in the agar, it's going to turn black. It's sort of a bluish black color. If the starch is gone, because it's been digested away by amylase, there will be a clear area on the plate. And that's what you're looking at down here in this image. Notice here on the left, this is a starch hydrolysis plate that has had iodine added to it. You can just barely see it, but there's a streak of yellowish growth right here down the center of the plate. So we took our inoculating loop and we made one long line of cells on the plate and then allowed them to grow. And after we added the iodine, we saw this strange result. On both sides of the growth line, the medium didn't turn black when we added the iodine. In fact, it went clear. We've got a piece of paper behind this plate because I wanted you to see that you can literally read through the medium. You can actually read the words. This medium has turned a clear yellowish, goldish color because there's no starch in here anymore. These cells secreted amylase, and the amylase broke down the starch in the area around the cells. So the cells could then absorb that glucose and use it. So this would be a positive result on a starch hydrolysis test. Now, in comparison, over here on the right, this is what a negative result looks like. Here's that streak line again. Here's the growth that grew over 24 hours. We added the iodine to the plate, and the entire plate turned black. There are no clear areas here. This is a negative result. These cells are not able to make amylase. They simply do not have the ability to produce that enzyme. So all the starch that's in this agar is still there. It has not been broken down at all. Whereas over here, it has. So this is what a starch hydrolysis plate looks like when you put the cells on it. It just looks like any other growth medium. It has that sort of beige color. We streak it with cells. We put it in the incubator. 24 hours later, we add the iodine to the plate, and it turns the agar black anywhere where there is still starch. Anywhere that the starch is gone, we will see clear areas. Make sense? It's an easy way for us to look to see whether or not those cells are making and secreting amylase. Now remember, amylase is only one of the exoenzymes in that big family of hydrolytic enzymes. It's only one of them, but it's a very helpful enzyme to have if you're a bacterial cell, because it allows you to harvest glucose from the environment. If you get into an environment where there's some starch around, you can harvest the glucose out of the starch if you're able to make amylase. Now, the other test we're going to be doing today is the OF test. And OF, again, stands for oxidative fermentative. This is going to test whether a microbe uses oxidative catabolism of carbohydrate or fermentative metabolism, or both. Remember, some microbes are able to do both. And we use a simple sugar in this test. And most of the time, including today, we're going to use glucose. Now, I want to say an important thing here, and make sure you put a star next to this in your notes. It is possible to use a different disaccharide in an OF test. For example, you might put in lactose, or you might put in maltose, or some other disaccharide instead of this monosaccharide called glucose. It's the glucose, remember, that's either going to be used for oxidative catabolism or fermentative catabolism. By putting in a disaccharide to start, you're also testing to see if the microbe can break apart that disaccharide to get glucose out of it. It's glucose that cells use for cellular respiration. It's glucose that cells use for fermentation. It's glucose. But remember, there are some disaccharides that are made out of glucose and another sugar. Lactose, for example, is made out of glucose and galactose. Sucrose is made out of glucose and fructose. So if we were going to use something other than glucose in an OF test, it would be because we wanted to know if they had the enzyme needed to break that sugar down to get at the glucose. Because it's the glucose that is used for the rest of the metabolic reactions, oxidative or fermentative. Glucose is the granddaddy of metabolism. Everybody uses glucose. It doesn't matter if you're a bacterium or a fungus or an archaeal cell or a plant or an animal. It doesn't matter. If you're a cell on planet Earth, glucose is the carbohydrate that you burn or oxidize in order to make ATP. Now, oxidative versus fermentative. Remember what these words mean. Oxidative has nothing to do with oxygen. When we use the word oxidative, we're talking about oxidation reduction reactions. In other words, can the cell use a series of oxidation reduction reactions to break down glucose? Or does it use a whole different process called fermentation to break down the glucose? Or is it capable of both? Oxidative does not have anything to do with oxygen. The word refers to redox reactions, oxidation reduction reactions. And remember, when it comes to breaking down glucose using oxidative catabolism, it can be either aerobic or anaerobic. We're really talking about cellular respiration when we talk about oxidative catabolism, cellular respiration. And sometimes cellular respiration is aerobic, and sometimes it's anaerobic. But both of those things are oxidative because both of those pathways use oxidation reduction chemistry. In aerobic cellular respiration, remember the final electron acceptor at the end of the electron transport chain is oxygen. In anaerobic cellular respiration, the final electron acceptor at the end of the electron transport chain is going to be something different. It's going to be nitrate or sulfate or carbonate, or some other molecule. Aerobic and anaerobic cellular respiration is oxidative. It uses oxidation reduction reactions in order to break down glucose. Aerobic or anaerobic cellular respiration is oxidative. Fermentation is a completely different set of reactions. So that's really what we're asking with the OF test. Is this an organism that's doing cellular respiration? Or is this an organism that's doing fermentation? Or can it do both? Now, because of the nature of this test, and it will make more sense in just a minute, but because of the nature of the OF test, we can only test for aerobic cellular respiration. So the OF test is testing oxidative versus fermentative. And for the oxidative, it's only testing for aerobic cellular respiration. So oxidative has nothing to do with oxygen. It's about oxidation reduction reactions. So whenever we talk about oxidative metabolism, we could be talking about aerobic or anaerobic. But for this test, it's aerobic that we're talking about. Remember, a lot of bacteria, in fact, most bacteria are facultative anaerobes. They prefer to do aerobic, oxidative cellular respiration. That's what they prefer to do. But if they don't have oxygen around, they can switch and do something else. There are some bacteria that can switch over and do anaerobic cellular respiration. There are some that can switch over and do fermentation. It depends. That's what we refer to as being able to switch to secondary metabolic pathways. Not the pathway you would choose, but if you have to, you can use it. That's your secondary metabolism. And the last thing to remember is this. There are some bacteria that only ferment. That's the only metabolic pathway they use to make their ATP. We call them pure fermenters. If you think about it, that's a tough way to survive on this planet. If you're a pure fermenter, all you can do is ferment. That's a tough way to survive. Because remember, fermentation is not a very good way to make ATP. It's not very efficient. You have to use a lot more glucose to get enough ATP in order to survive. Much better to use cellular respiration if you can, because it's much more efficient. You get a lot more ATP for each individual unit of glucose. But yeah, there are some bacteria that just ferment. It's all they can do. All right, on this slide, I've got a couple of key points. Key point number one, anaerobic cellular respiration is not the same as fermentation. Remember that. Fermentation is a completely different pathway. Anaerobic cellular respiration is simply cellular respiration that uses something other than oxygen at the end of the electron transport chain. It might use nitrate or sulfate or carbonate. Those are the most common. There are also some others, but those are the most common final electron acceptors. Two different metabolic pathways, not the same thing. Here's something else. Oxygen requirement categories, remember we talked about those? They describe the relationship that a microbe has with oxygen and the production of ATP through cellular respiration. Let me say that again, because that's important. When we used that thioglycolate broth, what we were looking at was the microbe's relationship with oxygen. Does it require oxygen? Does it require oxygen in order to do cellular respiration to make its ATP? That's an obligate aerobe. Does it find oxygen toxic? That's an obligate anaerobe. We went through all those categories. Here's the thing though. Oxygen requirement categories tell us nothing about whether or not a microbe can perform fermentation. It doesn't say anything about their ability to ferment. So you can't make any conclusions about whether or not an organism can ferment based upon which one of these oxygen categories it belongs to. You have to test. You have to test to see if it can ferment. Here's the third key point. Fermentation is a process that doesn't require oxygen. In other words, there's no step in fermentation where oxygen is needed. But it can certainly occur when oxygen is present. Oxygen is not going to stop it from happening, but oxygen is not required for fermentation to take place. All right, let's take a look at this OF test. OF medium is actually a basal medium. Remember what that means. That means it's chemically defined. Most non-fastidious bacteria will grow on it or in it just fine. OF medium takes this basal medium and adds a large amount of carbohydrate. And again, we can choose which carbohydrate we want to use, but most of the time we use glucose. We're going to use glucose today. OF medium also has a small amount of peptone in it. Now, peptone is a product of the digestion of protein. So peptone in the media is there to help support the growth of a microbe that cannot metabolize the sugar that we put in the medium under aerobic conditions. This is a confusing point, so let me say it again. In OF medium, we're going to put plenty of glucose, lots and lots of glucose, because we're testing to see, are they going to do aerobic cellular respiration, or are they going to ferment, or are they going to do both? They need glucose for both of those things. But remember, there are some microbes that can't do cellular respiration with oxygen. They need a different final electron acceptor. They need nitrate or sulfate or carbonate or something else. So for those microbes, if we put them into the OF test medium, they're in trouble. There's lots of glucose around, but if they only know how to do anaerobic cellular respiration, they're in trouble because they don't have their final electron acceptor available. And they're going to die. They're going to die unless we give them a secondary way to survive. That's what the peptone is for. The truth is, it is possible for cells, most cells, to use protein and amino acids to actually make glucose. Now that seems strange. That seems strange to us. It's not an ideal pathway. And cells don't do it unless they're absolutely forced to do it. Peptone, which remember is just partially digested protein, peptone gives them amino acids, which they can also catabolize or break down and use to make ATP. It's a terrible way to make ATP. It's not done unless the cell is dying, basically. So the peptone is going to allow these bacteria to survive during our test, basically during our test. But you're going to see it's going to give us a different result than what we would normally expect. Lots of carbohydrate in the medium, a little bit of peptone, just in case we ended up with an anaerobic cellular respiration microbe. It supports the growth of microbes that cannot metabolize this glucose under aerobic conditions. They just can't do it. They don't have the ability to do it. Now, here's a critical point about OF medium. OF medium contains one of these indicator dyes. And this one is called bromthymol blue. So this is a blue dye. Well, I shouldn't say that. It's a dye that turns blue under certain pHs, and it turns yellow in other pHs. All right. So if the medium is acidic, it will turn yellow. If the medium is alkaline, it will turn blue. If the medium stays neutral, it won't change color. You're going to see that the medium starts off green. So no change in pH, the media stays green. If the media turns acidic, it's also going to turn yellow. If it turns basic or alkaline, it's going to turn blue. Now, if the bacteria is metabolizing this glucose using either aerobic cellular respiration or fermentation, an acid is going to be produced as a product, and the medium is going to turn yellow. If we end up with one of those unusual instances where the bacteria can't use the glucose and it uses the peptone instead, the medium is going to turn blue. The peptone that gets metabolized is going to make the medium alkaline, and it will turn it blue. So this is a test where we have to interpret three colors. The medium starts green. If the carbohydrate gets catabolized, it's going to turn yellow. If the peptone gets catabolized, it's going to turn blue. Now, when we run an OF test, we actually run it in pairs. We take two tubes that have this OF medium in them, and we're going to inoculate both of them with our bacteria. One tube we're going to leave open to the air. This is going to test for aerobic, oxidative catabolism of glucose. In other words, this tube will show us whether or not the bacteria can do aerobic cellular respiration. Remember, cellular respiration is followed by that process called oxidative phosphorylation. That's what actually produces the ATP. With the second tube, we're going to inoculate it, but then we're going to put a little bit of mineral oil over the top of the medium. We're going to seal off the medium with mineral oil. What that's going to do is create an anaerobic environment. It's going to test for fermentation. What we're going to do is we're going to layer a little bit of oil over one of the tubes. Essentially, what we're doing then is we're preventing oxygen from coming into the medium. We're not removing oxygen out of the medium, though, are we? We haven't done anything to take away the oxygen. We're just preventing more oxygen from getting in there. Again, what this is going to do is it's going to force the microbe to ferment, if it can ferment. It's going to force it to ferment, if it can. So this is what OF tests look like. We always run them in pairs. So we always have two tubes of the medium. You can see I've got one pair here. I've got another pair here. And I've got another pair here. So all 6 of these tubes were inoculated and put in the incubator, inoculated with different microbes, obviously, because we have 3 different results here. And here's what we do. When we pull the tube out, we evaluate the color first. So when I see yellow in an OF tube, I think acid, acid. An acid has been produced. We're in acidic conditions in here. That's why it's yellow. First, you interpret the color. Then you would compare the open tube to the oil-covered tube. In the open tube, the acid was produced through aerobic cellular respiration. So this microbe has a positive result for aerobic cellular respiration. It can do it. This tube tests for fermentation, and I also got acid produced. So this microbe also ferments. It can do both things. The yellow color, in other words, makes me think, yes, positive for that metabolic process. Positive for cellular respiration in the open tube, positive for fermentation in the oil tube. Now look at this result. I got yellow in the open tube, but green in the oil tube. Remember, the medium starts green. So in this tube, I got a positive result. Yellow color means acid. An acid was produced. And that means that in this tube, the microbe did aerobic cellular respiration. But over here in the oil tube, it didn't ferment. If it had fermented, it would have produced an acid, and the medium would have turned yellow. So I've got a positive result for aerobic cellular respiration and a negative for fermentation. Over here, neither tube changed color. I've got one tube that's green and another tube that's green. It didn't do aerobic cellular respiration. It also didn't do fermentation. It didn't do anything. This organism probably died in here. Remember, there is some peptone in there. If the organism is able to use peptone to just survive, the media will go alkaline and it will turn blue. But this poor organism couldn't even do that. This poor organism couldn't even manage to do that. This is probably an anaerobic cellular respiration kind of microbe that couldn't survive. It could not use glucose aerobically for cellular respiration, and it could not ferment glucose. So I got left with two green tubes. So how do we interpret an OF test? Remember, we're going to inoculate 2 tubes of OF medium. One tube we're going to leave open to the air. The other tube, we're going to layer a little bit of mineral oil on top to prevent any more oxygen from getting in. We're going to put both tubes into the incubator for 24 hours. And when we pull the tubes out, we're going to do 2 things. First, we're going to evaluate the color changes, if any. And then we're going to compare the open tube to the oil tube. If the tube turns yellow, we know we're in acidic conditions. Growth is occurring in there, and the glucose is being catabolized. It's being broken down. That's what yellow means. Remember, we're just thinking about color here. Yellow means acid. Those cells are growing and metabolizing and producing an acid. That means they're breaking down the glucose in there. If the tube stays green, that's no change. The pH has not changed. There's no growth occurring in there. There's no metabolism of glucose going on. There's also no metabolism of that peptone going on. Those cells are not growing. They're not metabolizing. If we see blue, we know we're in alkaline conditions. Growth is occurring in that tube, but there's no metabolism of glucose going on. Those cells are metabolizing peptone just to survive. They would not be able to survive very long on just peptone, by the way. but it will keep them alive long enough for us to know they're in there and they're growing. They're just not using the glucose. That's how you evaluate the color. Then compare the open air to the oil. In the open air, remember, we're looking at oxidative catabolism of glucose. In other words, aerobic cellular respiration for this test. In the oil-covered tube, we're looking at fermentation of glucose, fermentative catabolism of glucose. Remember, in lecture, we talked about how fermentation produces one of three products or some combination. Fermentation can produce an acid, it can produce alcohols, it can also produce gases. So depending upon the organism and the pathway of fermentation that it's using, because there are many, you're either going to get acid, alcohol, or gas, or some combination of those. Any acid that gets produced in that tube is going to turn that tube yellow. It doesn't matter what kind of acid gets produced, the tube's going to turn yellow. Several different acids can be made during fermentation. It depends. It depends on what sugar they're fermenting and the organism you're talking about. So when you see yellow in the oil-covered tube, all that you know is that fermentation is occurring. You don't know what kind of fermentation is occurring. You can say that organism ferments glucose, but that's all you can say. You can't say which pathway of fermentation it's using. Remember from lecture, there are lots of different pathways for fermentation. Lots of different pathways and lots of different products. Some of those products we actually use commercially. But yeah, all you can say is, yeah, it's fermenting the glucose. It's fermenting the glucose, but I don't know what pathway it's using. The other thing to recall is this. Remember, oxidative metabolism, again, for this test, we mean aerobic cellular respiration. That's also going to make an acidic condition in the tube. And it's also due to acid. But you might say to yourself, hold on a minute, what acid is made during cellular respiration? Remember the three processes of cellular respiration. Glycolysis is first. That takes glucose and makes pyruvate. It breaks some of the bonds in glucose and it makes pyruvate, glycolysis. The second set of reactions, that's called pyruvate processing. The cell takes that pyruvate and breaks it down a little more into acetyl-CoA and carbon dioxide. The third set of reactions is called the Krebs cycle. The cell takes that acetyl-CoA and finishes breaking it down. Breaks it down all the way to carbon dioxide. By the end of the Krebs cycle, all of the glucose has been broken all the way down into carbon dioxide. All of the electrons that got released during that process, they get sent down the electron transport chain. Their energy gets harvested. And they land on, at the bottom of the chain, they land on and combine with oxygen to make water. That's the end of cellular respiration. We still have to do oxidative phosphorylation to make the ATP, but cellular respiration ends at the bottom of the electron transport chain. So you may say to yourself, well, where's the acid? Where's the acid in that? Here's the answer. Remember, glycolysis takes glucose and makes pyruvate. Pyruvate processing takes that pyruvate and makes acetyl-CoA and carbon dioxide. The Krebs cycle takes the acetyl-CoA and makes carbon dioxide. So by the end of the Krebs cycle, all you've got left is carbon dioxide from the glucose. Carbon dioxide in a living cell is going to combine with water. And it's going to produce a molecule called carbonic acid. There's your acid. So in that open air tube, if the cell, the microbe, is using aerobic cellular respiration, the tube is going to turn yellow because all that CO2 that's being made is going to be converted to carbonic acid. And we're going to end up with a yellow tube. And that's how we know it's doing cellular respiration. All right, this is a good place for us to take a break. Let's take a 5-minute break. I'll see you back at 10:05. Okay, everybody, when you come back in, just type your name into chat for me. Thank you, I appreciate that. Yeah, just type your name into the chat for me, and I'll have a record that you were here. Very good. All right. So let's go through a summary of what we just talked about before we look at our experiment. When we do an OF test, we're going to be interpreting 2 test tubes with this OF medium in it, one that's open to the air and one that's covered with oil. We have to be able to interpret 3 color changes on this test, because we may get tubes that have turned yellow, we may get tubes that have stayed green, and we may get a tube that's turned blue. Remember, in the yellow, we know that there's growth going on. We know the cells are dividing. They're also metabolizing glucose, and they're producing an acid. That's what the yellow color tells us. And then we have to compare the open tube to the oil tube in order to know which pathway they're using. In green media, there's no growth going on. Those cells are not growing and dividing. Those cells are not metabolizing the glucose. There are some bacteria that simply cannot grow in OF medium. Question is, why? Why? Why would a bacterium not be able to grow in OF? What do you know about OF medium as a medium that might explain to you why a bacterium simply can't grow in it at all? It can't grow in the open tube. It can't grow in the closed tube. Why? Can't grow at all. What do you think? What kind of medium is OF? What category does it belong in? Go back in your notes. What kind of medium is OF? Is it selective? Is it complex? Is it differential? What kind of medium is it? Let's take a look. OF medium. It's a basal medium. It's basal. What does that mean? What does that mean to us? Type it into chat. There are some bacteria that cannot grow in OF because it's a basal medium. What kind of bacteria can't grow in basal media? Chemically defined media. What do you think? That's a good guess, Lisa, but remember, this is a bacterium that doesn't even grow at all. It doesn't grow with the open air. It doesn't grow with the oil covered. It won't even grow with the peptone. Why? It's because it's a basal media. What do you know cannot grow in a basal, chemically defined medium? Candy's got it. Good. That's right, Candy. That's right. Fastidious organisms can't grow in basal media. You've probably got a bacterium that has some strange little metabolic requirement. It has some need for some ingredient, some bizarre ingredient that's not in that medium. So this isn't even about testing its metabolic abilities. This is about the fact that this is a basal medium. It's chemically defined. And that means some bacteria just can't grow in it because they're fastidious, because they need extra special complex ingredients in order to grow. So if you see green tubes, you got no growth. It didn't grow. And what that should say to you is that, oh, that bacterium just couldn't manage to grow at all in here because it has some special nutritional requirement that's not met by this medium. It's not that it couldn't use the glucose. It couldn't use the glucose. It couldn't use the peptone. It couldn't grow at all because it didn't have a nutritional ingredient it needed. That's what a green tube is. Okay. So in green media, there was no growth. There was no metabolism at all. No metabolism of glucose, no metabolism of peptone. Some bacteria just can't grow in OF because they're fastidious. Now, if you get a blue tube, you've got growth. They're growing and they're metabolizing, but they're not metabolizing the glucose. They're metabolizing the peptone. And they're doing it just to survive. It's not ideal. They're using a pre-digested protein just to survive. It is not an ideal environment for them. They're not going to live very long. But it allows us to see the difference between no growth, no metabolism, and no metabolism of glucose. Okay, we have to be able to tell the difference between those conditions. Good. All right, here's our experiment. We're going to use 3 bugs today. 2 of these we've already seen. One of them is new today. We're going to use our old friend E. coli, gram-negative rod. Remember from our oxygen category lab, we learned that E. coli is a facultative anaerobe. Facultative anaerobe. So can you predict what kind of results you might get on OF? This is a facultative anaerobe. Remember, on our oxygen, our thioglycolate test, that means it grows best in the oxic zone up at the top. It can also grow throughout the rest of the tube. So it grows best in oxygen. It can even grow with no oxygen, and it can grow in any low level of oxygen. Facultative anaerobe. So can we predict how it's going to do in the OF test? What about the open air tube? Remember, that one is testing for aerobic cellular respiration. What color do you think we're going to get in that tube with E. coli? Type it into chat. What color change are we going to see in the open air tube for a facultative anaerobe like E. coli? Is it going to be yellow? Is it going to stay green? Is it going to be blue? Lisa's got it. Lisa jumped right in. Good girl, you got it. Candy's got it. Maddie's got it. Sop's got it. Good. Yellow. Yeah. This is an organism that loves to do aerobic cellular respiration. It's its preferred pathway. It can do other things though too, right? It can grow in no oxygen, and it can grow in low levels of oxygen. Okay. What about the oil-covered tube? Can a facultative anaerobe ferment? Yes or no? Or I don't know. Lisa, you're on fire today. Yes, no, or I don't know. Lisa's got the right answer. Maddie's got it. Yeah, I don't know. I can't make that prediction, can I? Oxygen category tells us nothing about ability to ferment. Oxygen category tells us nothing about ability to ferment. We can make a great guess. Yeah, we can make a guess for the open tube, but we cannot make a guess for the oil tube just knowing its oxygen category. Okay, remember that. That seems like a great question, doesn't it, to put on an exam? Or I should say a lab practical exam. Yeah, you cannot make predictions about fermentation based on oxygen category. That's part of the reason we have to do these tests. All right, so we've got E. coli. Our second organism is Bacillus subtilis. We saw this organism earlier in the summer. This is a gram-positive rod. The organism we haven't looked at really yet is Pseudomonas, Pseudomonas aeruginosa. This is a gram-negative rod. Pseudomonas is a fascinating bacterium. This is a bacterium that you frequently encounter in environmental testing. It loves dark corners, wet, dark corners of buildings. It loves to grow in hospitals and doctors' offices. Pseudomonas is what we call an opportunistic pathogen. Pseudomonas is a terrible problem in patients who are severely compromised. My favorite example is burn patients. Burn patients can pick up Pseudomonas from the environment if the people who are caring for the burn patients are not very, very careful. They can accidentally introduce Pseudomonas onto the surface where skin is missing. And Pseudomonas will take advantage. It's opportunistic. It takes advantage of an opportunity that normally isn't there. So in a burn patient, they can get a Pseudomonas infection. And it's horrible. It's a horrible infection. And the reason that you don't see Pseudomonas infections in you and I, in our skin, is because our skin keeps it out. But if you don't have skin because you're horribly burned, Pseudomonas can take advantage of that. And it can start to grow in the deeper tissues. And yeah, it's a really bad infection, Pseudomonas. You can get Pseudomonas infections in people who are severely compromised with other problems. You know, they're in the hospital for other problems and they're very sick. These are very sick people. Pseudomonas will take advantage of that. It's opportunistic. It looks for an opportunity to get into the body because the person's compromised. All right, so those are our three organisms. We're going to do two tests. We're going to do the starch agar. We're going to use a simple streak. So just one streak with the inoculating loop down the plate. And we're going to look for growth after 24 hours of incubation. Remember, then we're going to apply iodine to the agar. And we're going to look for evidence of starch hydrolysis. So 1 plate for each of our 3 organisms. For the OF medium, we're going to run a pair of tubes for each organism. Both tubes will be inoculated. One will be left open to the air and put in the incubator. One will be covered with mineral oil and put into the incubator. And we will check those tubes after 24 hours of incubation. Take a look at this slide. This is OF medium. This is the recipe for OF medium. Here's the peptone. Now, these amounts aren't going to mean a whole lot to you, but you can make a comparison between the amount of glucose in this medium and the amount of peptone. There's 5 times as much glucose in here as there is peptone. There's a little bit of salt, not too much, just a little. Here's that bromthymol blue. That's our indicator dye. And we also have dipotassium phosphate as needed for these organisms. Very simple medium, isn't it? It's a very simple recipe. This is a basal medium. Now, here's my question for you. Are there any final electron acceptors in here that a microbe could use for anaerobic cellular respiration? Anaerobic cellular respiration. If you were a bacterium and that was your preferred pathway and you got placed into this medium, can you do anaerobic cellular respiration in here? What do you think? Remember, nitrate, sulfate, carbonate, 3 most common, with nitrate being the most common. You see any nitrate in here? See any nitrate in this medium? There's no nitrate in here. There's no sulfate in here. Boy, this is a basal medium, right? Talk about a simple medium. No, there's no final electron acceptor in here that an anaerobe could use. The potassium, the dipotassium phosphate, that's a hard one to say, has potassium and phosphorus and oxygen. Dipotassium phosphate. No nitrate, no sulfate, no carbonate. An anaerobe that does cellular respiration can't do that in this medium. It's just not there. So here's a question. Oh, hold on. Lisa's got a question. It's bound together and the phosphate can't be used. The phosphate can absolutely be used, but nitrate is nitrogen and oxygen. Sulfate is sulfur and oxygen. Carbonate has carbon and oxygen, and I can't remember its formula off the top of my head. So yes, that phosphate could certainly be used, but all that is phosphorus and oxygen. So we don't have a final electron acceptor in this mix. And what that means is this test, this OF test, cannot test for anaerobic cellular respiration. It just can't, because there's no final electron acceptor for them. Why can we test for aerobic? Where does the oxygen come from? Why can we test for aerobic cellular respiration? Where does the oxygen come from in that open air tube? Yeah, Lisa's got it. It comes from the air. It comes from the air. It comes from the atmosphere, right? It's an open air tube. There's no auger in that tube to impede the oxygen from coming in. So the oxygen is literally just going to diffuse down into that open air tube. And that's why that tube tests for aerobic cellular respiration, not anaerobic. Good job. All right, let's take a look at our results. Starch hydrolysis test. Here's the E. coli plate. We pulled it out of the incubator. You can see growth along this streak line right here. This was just a simple streak, back and forth, back and forth. We put iodine on the plate. Here's the result. Here's Pseudomonas. On this plate, we didn't even go back and forth. We just made one long straight line. Here's Bacillus subtilis. 3 plates. E. coli, top left. Pseudomonas, bottom left. Bacillus subtilis on the right. Remember, when we read a starch hydrolysis test, we're looking at the media. We're looking at the media. What do you see? What do you see for E. coli, positive or negative? Is this a positive or a negative test? Cells grew on it. Cells grew on it. What do you think? Lisa's got it. Maddie's got it. Candy's got it. Good. Negative. The media stayed black. The media stayed black. Remember what black means. The iodine is going to bind with any starch that's in the plate and turn it black. So anywhere that you see black, you're looking at starch. All of the starch on that plate is still there. It has not been broken down by any amylase. So the E. coli plate is negative. So is the Pseudomonas plate. Here's the growth line. All the rest of the plate is black. All of the starch is still there because it bound with the iodine and turned black. But look at the Bacillus subtilis. The Bacillus plate, the media has cleared out around the growth lines. It's this clear, sort of a goldish color. In other words, there was no starch here for the iodine to bind to. So you got no black color here. Bacillus subtilis had a positive result. Bacillus subtilis makes and secretes amylase. That's one of its traits. It's one of its features. It is able to make and secrete amylase, and it will break down the starch that's around it into glucose, and then it will absorb that glucose and use it. So here's a question. If I took a little bit of that agar out, if I scooped a little agar out in the clear areas, I wouldn't find any starch in there. Would I find any carbohydrate in there? If I scooped out some of the clear agar and took it to the lab and tested it for carbohydrate, would there be any carbohydrate in there? There's no starch in there. What carbohydrate, if any, would be present in those clear areas? What's starch made out of? What's starch? What is starch? Austin's got it. Yeah, there'd be all kinds of glucose in there. Remember, the amylase is going to break down the starch into its glucose. That's all starch is lots and lots of glucose bound together. The amylase is going to break the bonds between the individual glucose molecules. And you're going to find a lot of glucose in that clear area. Now, the cells are going to start absorbing as much of that as they can, but there'll be a lot more glucose that is left over in those areas if you were to test for it. Good job, Austin, Maddie, good job. All right, here are our OF tubes. On the left, this is E. coli. In the middle, this is Pseudomonas. And on the right, this is Bacillus subtilis. I see a couple things here we haven't seen yet. I see some blue. We haven't seen blue yet. There's your blue color. I see a tube that is green and yellow. Holy smokes, we haven't seen that yet. Down here on the bottom, these are your references, just, that we looked at, so you can compare. Remember, this is the double yellow means one thing. Single yellow, green, double green. We talked about these a moment ago. Let's start with E. coli, because that one's pretty obvious. Remember, evaluate the color first. I've got yellow. I've got yellow color. What does that tell me? Is anything growing in those tubes? Yes or no? Is anything growing in a yellow tube? Type it right into chat for me. Is anything growing in those tubes that turn yellow? Sarah says yes. Maddie says yes. Sop says yes. And an acid was formed. Exactly. Yeah. Yellow means growth occurred and the glucose was catabolized. It was broken down. Growth occurred and the glucose was used. It was broken down or burned or oxidized or catabolized. Good. That's what yellow means. Now, compare the two tubes. Open air, what pathway must have happened in that open air tube? What pathway produced that acid in the open air tube? Be as specific as you can. Open air tube turned yellow. That means growth is occurring and acid was produced. Good. Lisa, good. Sop, Candy, Maddie. I almost called you Candy. Good. Aerobic, cellular respiration. Good. Now the oil covered tube. It too turned yellow. Yellow means growth occurred, acid was produced. What pathway? What general pathway? Because we can't be specific. What general pathway occurred in that oil covered tube? Good job, Sarah. Fermentation. Good job, Maddie. Fermentation. We don't know what kind of fermentation it was, but we know it was fermentation. It's yellow. That means growth occurred and acid was produced. And because it's the oil covered tube, we know it was fermentation. We blocked additional oxygen from coming in. So we forced that microbe to ferment. E. coli is a facultative anaerobe. It loves to do aerobic cellular respiration. That's its favorite pathway. But it's also capable, it turns out, of fermenting. What an adaptive organism E. coli is. All right, let's look now at the bacillus. Remember, evaluate the color first. I've got blue and green in the open air tube, and I've got green in the oil tube. Color first. What does blue mean? What does blue mean? Is anything growing in there? You're right, Candy. Alkaline, alkaline. Is there growth in that tube? Austin says yes. You're right, Austin. Yeah, something's growing in that tube, and that means it's metabolizing. Is it metabolizing the glucose or something else? It's not metabolizing the glucose, because if it did, it would make acid. Whatever it's metabolizing is not making an acid, it's making it alkaline in there. It's metabolizing the peptone. It's metabolizing the peptone. And that's why the pH went alkaline, higher than 7. Basic is another way to say that. The peptone is being metabolized by those cells. That tells us something. That tells us that this organism cannot catabolize the glucose in the open air tube. But it can grow in there. I mean, it's growing and it's using the peptone to survive, because it doesn't have what it needs to use the glucose. So it's blue. Now, you'll notice that only the top half of that tube is blue. The whole tube isn't blue, only the top half is. In these cases where you get a blue color, remember, those are cells that they're stressed. They're using a secondary metabolic pathway. They're trying desperately to survive. They're not growing well. They're not growing well. That tube may be, when you see this kind of a situation, you may get a tube that's all blue, but it's more common that it's only partially blue. They're growing, but they're not growing very well. They're stressed out. All right, how about this weird oil covered? Well, actually, we're still on bacillus. We're still on bacillus. The oil covered tube for bacillus is green. Green, 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 top to bottom. What does that tell us? Is there anything growing in that tube? Do I have cells growing in that oil covered tube? What do you think? No, I don't have any growth in there. No metabolism in there. No change in pH, either acidic or basic. No change. Can that organism ferment? Can Bacillus subtilis ferment glucose? Because that's what I'm looking at in that tube. I'm looking at its ability to ferment. Can it ferment glucose? Is it fermenting the glucose in that tube? No. No. So Bacillus subtilis is an interesting organism, and at least from what we've been looking at. This is not an organism that's just jumping in and doing aerobic cellular respiration, and it's also not able to ferment the glucose in the tube. So, it's very different from E. coli. It's a very different organism. Now, you'll notice that I'm not interpreting it by saying, oh, it must be an anaerobe. It must be doing an anaerobic cellular. It must like to do that. You'll notice I'm not saying that. The reason I'm not saying that, even though that's what's left over, is because this is not a test. I am not testing whether or not it can do anaerobic cellular respiration. The OF test tests for aerobic cellular respiration or fermentation. So all I can really say from an OF test is it does not do aerobic cellular respiration and it does not ferment glucose. That's all I can say. There are other tests that we will do to confirm if it's an anaerobic cellular respiration type of organism. Okay. Now let's look at Pseudomonas. Look at the oil-covered tube first. Green top to bottom, looks just like the oil-covered tube for Bacillus. Pseudomonas cannot ferment glucose. Look at this strange open air tube. It's yellow at the top, green at the bottom. Now, I know it looks almost blue down here, but it really wasn't. It's really green all the way down. So yellow at the top, green at the bottom. This is a weird looking tube, I know. And it seems strange, like how can it produce an acid, but only produce some acid, not a lot of acid? What's going on in that tube? Pseudomonas, I'm so glad you typed that in, Lisa. Could this be like the peptone example? Could this be that these cells are metabolizing glucose, but they're barely surviving? Remember, in the reason that the bacillus open air tube was only partially blue is because we said those cells were growing really poorly. That's not the case with Pseudomonas. If a cell or a microbe is able to do aerobic cellular respiration, it's going to grow great in an open air tube. It has tons of glucose to work with. So if it's able to do it, it's going to grow fine. The weird thing about Pseudomonas is it grows very slowly. It's growing according to the way it grows. But Pseudomonas just happens to be known as a bacterium that grows very slowly. In other words, if I put some Pseudomonas on an agar plate and I pulled that agar plate out after 24 hours, I'm not going to get great growth. You have to leave it in a little longer. Pseudomonas needs about a day and a half or even 2 days in the incubator. It's just one of its characteristics. It just grows slowly. So it's not that it's having trouble growing in that open air tube. It's not, it's growing at its normal slow rate. So it's making acid slowly. And that's why only part of the tube turns yellow. If you were to put that tube back in the incubator for another day, the whole tube would be yellow. That's just a little idiosyncrasy for Pseudomonas. It just grows slowly. So Pseudomonas is positive for aerobic cellular respiration. It's just growing slowly. It's negative for glucose fermentation. This is a slide that came from another lab. This is pairs of OF tubes that have all been inoculated with Pseudomonas. This pair of tubes was left to incubate for 24 hours. This one was left to incubate for 48 hours. This one was left in the incubator for 5 days. Now, in my experience, you don't need to leave it for 5 days, but notice the open air tubes. We got a little bit of yellow after 24 hours. We got more yellow after 48 hours. The whole tube is yellow after 5 days. These cells are growing just fine. They're not having any trouble growing. There's lots of glucose in there, and they can catabolize that glucose. They can use aerobic cellular respiration to break down that glucose. And that's exactly what they're doing. They just grow very slowly. So it takes them longer to divide. So they make less acid after 24 hours than, say, E. coli would, because E. coli grows quickly normally. Pseudomonas is just a slow grower, as we say. So yeah, it doesn't, in my experience, it doesn't take 5 days, but who knows, maybe that strain, it did take 5 days for the whole tube to go yellow. So when you look at OF tubes, remember, look at the color first. Evaluate the color first. Then compare open versus oil covered. For color, if it's yellow, it means acidic conditions, growth is occurring, and the cells are catabolizing the carbohydrate, which is glucose. If it's blue, it's alkaline. Growth is occurring, but it is not catabolizing the glucose. Instead, It's breaking down the peptone in order to survive. Green tubes mean no change in pH, no growth is occurring, no metabolism of glucose is occurring, no metabolism of peptone is occurring. It just can't grow in there. And that may be due to the fact that it's fastidious. Once you've evaluated the color, then compare. Open, if it's yellow, you've got an organism that can do oxidative catabolism of glucose. Specifically, it can do aerobic cellular respiration. In the oil tube, if it turns yellow, you've got fermentative catabolism. We don't know which pathway it's doing, but we know it's fermenting the glucose. So E. coli can do both. E. coli can do aerobic cellular respiration. It can also ferment. Remember, oxygen category cannot tell us anything about fermentation. All it tells us, oxygen category, is its relationship with oxygen. E. coli is a facultative anaerobe. That means it prefers to do aerobic cellular respiration, but it can also grow in low oxygen and no oxygen. All we've done here is we've pointed out that it can do aerobic cellular respiration and it can do fermentation. We cannot say anything about anaerobic anything here because we didn't test for it. Pseudomonas can use aerobic cellular respiration to catabolize glucose, but it grows slowly. It cannot ferment glucose. Bacillus subtilis, it cannot catabolize glucose under these conditions. Instead, it catabolizes the peptone. So it cannot do aerobic cellular respiration, at least in this medium. Over here, it also cannot ferment. I cannot make any statements about its ability to do anaerobic cellular respiration because I didn't test for that with OF. So in our experiment, which organism secreted amylase to digest the starch? That was Bacillus subtilis. Bacillus subtilis secretes amylase and can break down starch in its environment. And then it can bring the glucose in. It can absorb the glucose and use it for production of ATP. Which organism can aerobically oxidize glucose? Or we could say catabolize glucose using redox reactions. Or we could say can use aerobic cellular respiration. Remember, all of those mean the same thing. That would be our E. coli and our Pseudomonas. Both of those organisms produced a yellow color in the open air tube. And that tells us that they can metabolize glucose using oxidative pathway, aerobic cellular respiration specifically. Which organism can ferment glucose? We only had one, that was E. coli. Which organism can grow but cannot catabolize the glucose in this particular medium? That's our bacillus. Bacillus can grow, but it can't use the glucose for aerobic cellular respiration. It just can't. It's using peptone in order to survive. It can do that, but it's just not able to use aerobic cellular respiration in this particular medium anyway. Look for the clear area around the growth. That clear area tells you that you've got a positive test for starch hydrolysis. Remember, there are lots and lots of hydrolytic enzymes. There are exoenzymes like amylase. There are also endoenzymes. This test looks specifically for an exoenzyme called amylase. It secretes the amylase out into the environment, and the amylase breaks apart the bonds between the glucose molecules, breaks down the starch into glucose so it can be absorbed. In OF tests, we always run a pair of tubes, one open to the air, one covered with mineral oil. We evaluate the color first, and then we compare the two tubes. OF testing tests for the ability to do aerobic cellular respiration and the ability to ferment glucose. It doesn't test for the ability to do anaerobic cellular respiration. So we can't make statements about anaerobic until we test for it specifically. And we will do that. We will do that in a little bit later test next week. Very, very common test, the OF test. Now, like I said at the beginning, we put glucose into the OF test most of the time because glucose is the universal carbohydrate. It's the carbohydrate that all cells use to make their ATP, regardless of which pathway that they use. Some organisms are able to take another sugar, a bigger sugar, like a disaccharide, and they can break it into its individual monosaccharides, one of which happens to be glucose. So it is possible, if you want to know if a bacterium has the enzyme, for example, to break apart sucrose, or to break apart maltose, or to break apart fructose, or to break apart any disaccharide, lactose. If you want to know, you can certainly put a different sugar into your OF test. You can also run other carbohydrate catabolism tests besides OF. It's just that OF is a very nice basic test. It's a very simple, basic, easy to perform test. And by running the OF, we get a very clear answer about whether or not a microbe can do one of these two pathways. That gives us more information about that microbe. More information that can help us identify it. All right, very good. Any questions? All right, when we get together on Wednesday, we're going to look at a different test. We're going to look at fermentation specifically. We're going to look at a test that allows us to investigate fermentation a little more specifically. So, for example, if I did an OF test and I got a result like I got for E. coli, telling me that E. coli ferments glucose, I may want to investigate E. coli's fermentative abilities a little bit more. And we'll look at some testing that does that when we get together on Wednesday. All right, everybody, if there are no other questions, that's all I have for you today. I hope you have a great rest of the day. And I will see you on Wednesday. Bye, everybody.